The overall goal of this experiment is to determine the optimal chromosomal position of any given DNA element using transposon-mediated random insertion, competition experiments, and whole genome sequencing.
This method can help answer key questions in the DNA replication field, such as the importance of a chromosomal location of a genetic element. The advantage of this technique is that it couples transposon-based random insertion with the selection of the fittest clone by growth advantage, here exemplified by the E. coli DAS2 region.
Though this method can provide insight into the chromosomal organization of Escherichia coli, it can also be applied to other bacteria.
After preparing electrocompetent cells, such as Mg1655 delta DARS2 according to the text protocol, add 1 microgram of PNK bore DARS2 to 40 microliters of cells, and transfer the mixture to a pre-chilled sterile 0.2 centimeter gap cuvette. Insert the cuvette into the electroporation chamber, then electroporate the cells at 18 kilovolts, 500 ohm, and 25 microfarad. The time constant should be about 5 milliseconds, and no arcing should occur. Quickly recover the bacterial suspension by resuspending it in 1 milliliter of pre-warmed LB broth, and transfer to a 15 milliliter test tube. Incubate the cells under aerated growth conditions at 37 degrees Celsius for 30 minutes, without antibiotic selection. Then plate the bacteria onto the LB agar plates supplemented with 50 micrograms per milliliter of kanamycin, and incubate the plates at 37 degrees Celsius overnight.